Third time's a charm. First one, no audio. Second one, the slides would not advance. So let's hope we can get through this. I'm going to do my best to get this done efficiently without rushing, but it does take a while for the video to upload. So I have to record it, and then it's going to take time to upload, but I will post it as soon as humanly possible. We're talking about the non-specific immune responses, and that doesn't mean random. It means that the immune system is recognizing that something that does not belong in the body is present. It doesn't know that it's a specific bacterium or virus or protist, but something that does not belong in the body. Varicella is listed here. It is a virus. It is the one that causes chickenpox. It is also called herpes zoster because it is in the herpes virus family. And like all other herpes viruses, once you have contracted this virus, it never leaves your body. It will be with you for all time. And also, like other herpes viruses, the chickenpox virus can be reactivated under stressful conditions. It reactivates as shingles. It is super contagious, and it is spread by respiratory droplets, even though it shows up on the skin. And this crusty rash that's extremely itchy that shows up is because of the host's immune response to the presence of that virus. And inflammation is part of our innate immunity. All of the forms of innate immunity, the non-specific immunity in this chapter, are ones that we are essentially born with, and they do not change or adapt over time. So if you see the same organism more than once, your innate immune system reacts in exactly the same way every time. The adaptive immunity, which we see in chapter 18, does change, but innate does not. It will always have the exact same response no matter how many times it sees the same pathogen. Our skin is our first line of defense, and our tissues have various ways of sticking together that prevents microbial spread. And for this chapter, all you really need to know that there are 3 different ways that our tissues are stitched together. There's the tight junctions, the desmosomes, which is like a shoelace going back and forth to link those 2 tissues together, and the gap junctions that are like an iris diaphragm that open and close. And that's what can prevent the spread of microbes from one cell or tissue to the next. This is your exactly 1 slide anatomy and physiology lecture on the skin, because our skin is our first line of innate immunity. It is dry. The top layer of our skin is dead cells, and they flake off. So even if pathogens are attached, we wash them off, rub them off all the time. However, we do have hair follicles, and that is a way that microbes can evade this innate immunity. Sweat glands are another opening in there, and our sebaceous glands are all ways that they could get past that top layer. We have an example here of the Rose Gardner's disease because it happens most commonly with Rose Gardner's, but it's not limited to that. It happens because scratches, cuts, or punctures from the thorns on the rose bush introduce the fungus underneath the skin. So if our skin is cut or scraped or punctured, then we have breached that stage of innate immunity, and then different types of immunity are going to come into play. Our respiratory system is full of ciliated cells that act like little scrub brushes. So sneezing and coughing is an irritation, and the cilia that have with their mucus trapped dust particles, microbes, viruses, then when we cough or sneeze, those are expelled out. But those ciliated cells don't recognize a specific species of bacterium or a specific family of viruses. It's just this particle doesn't belong and gets swept away. the mucus cells produce that sticky substance that traps a microbe long enough for us to cough or sneeze and expel it. And we don't have to know the anatomy and physiology of a mucus-producing cell, but this one is in our intestinal epithelium. We have mucus-producing cells in our respiratory system, in our digestive system, and even in our reproductive system to create that sticky substance that will trap the microbe before it can get deeper into our tissues. In a previous lecture, we saw entry and exit portals for microbes, and those are also going to be places where our innate immunity can help us out. So microbes could evade our innate immunity, but our eyes have several different ways to protect us. Blinking is a mechanical means of trying to get the microbe off our eye surface. Our tears are liquid that can help wash the microbe away, which is also a physical means of removal. But our tears also have lysozyme. It's an enzyme that lyses microbes, in particular bacteria. All of those things help to protect and prevent microbes from getting into our tear duct and getting established and colonizing and causing infection. Our urine also is a way of washing our system. Our bladders are a large storage system. The urethra is very narrow, and that creates a high pressure for the urine to exit our bodies, and so mechanical flushing to keep it clean. But the urine is also an acidic pH, and that prevents microbes from growing. Again, our hair follicle, the skin is not 100% tight up against the hair shaft, so microbes could get into there, and if they do, they can even get into deeper tissues and into the bloodstream to spread. The sebaceous gland produces sebum, so our resident microbes eat that, and that gives them an incentive to stay with us. They take up space to prevent pathogens from landing on us. When they eat that sebum, they also produce oleic acid, which is acidic, and it's another means, a chemical means, of preventing pathogens from establishing themselves. So a pathogen would have to evade both the skin up against the hair shaft and the oleic acid from the native flora, eating that sebum and producing the acid to lower the pH. So put me somewhere else. The complement system is called that because it acts together with more specific and adaptive portions of our immune system. It complements it. You don't have to know all of the order of these complement pathways and the order of the numbers of the chemicals that are triggered, because the goal, you can see here, is to get to this membrane attack complex. The complement proteins 6, 7, 8, and 9 bind to the pathogen surface, attack its membrane, and destroy it, explode it. There are different ways that these complement proteins can be triggered to respond, and the classical pathway is with antibodies and antigens bound together. Antibodies are part of our specific adaptive immune system. They are little fork-shaped proteins on the surface of B cells, and antigens are any substance that generate an antibody response. So when our antibodies bind to antigens, and that is a specific adaptive response, it actually triggers this complement system. So the complement system, these series of proteins, they will respond in exactly the same way no matter which microbe has been found. Once the specific part of our immune system finds it, they will always act in the same order to trigger this binding to the membrane and exploding or lysing the pathogen. Just can't find a good spot for me. There we go. Cytokines are molecular messengers. And in particular, we're talking about in immunity. There are all sorts of other chemical signals that do other things in our cells. And they are also autocrine, paracrine, or endocrine signals. But we are discussing in this chapter just cytokines that are triggering immune responses. So autocrine means self. So the cell secretes a signal, and that cytokine signal signals back to the cell that made it. So it's a self-signaling. Paracrine means nearby. So this cell secretes a signal, a cytokine, and that triggers a nearby immune cell that's got receptors on its surface for that cytokine. Endocrine means it has to travel through the bloodstream or through the lymphatic system to a very distant site to have its activity. And the B cells we're going to see in chapter 18, they produce the antibodies. Interferons are cytokines, so they are a chemical messenger involved with immunity, and they interfere with viral replication. This cell has been infected by virus. That infection triggers the cell to release interferon, and it can act in 3 different ways. It will signal nearby cells to destroy RNA, and if you don't have RNA, you can't get translation. If you don't have translation, you don't have proteins, and that cell is going to die, but it's also not going to be infected by virus. It can also signal neighboring cells that have already been infected to undergo apoptosis. That is programmed cell death. So this is a natural occurring system in our cells. In this case, it's for a viral infected cell will be killed, and that will prevent this virus from spreading to other cells. But apoptosis is used for other things. If we have damaged cells or tumor cells, they are often triggered to undergo apoptosis, programmed cell death. And interferon also activates other immune cells that are specific to go out and be recruited to where the viral infection is happening and help to prevent its spread. This slide is showing the history of production of all of the formed elements in blood, meaning the cells in blood, because our blood has the fluid portion, the plasma, and then all the cells. They are called hematopoietic stem cells, meaning they are stem cells, and they can become any kind of blood cell. They can't become any kind of cell in our body. So all of our blood cells are formed in the bone marrow. Other types of stem cells could form other categories of tissue. The only kind of stem cell that can produce any kind of cell is an embryonic stem cell, and those happen in the pre-implantation stage. So when we are a fertilized egg, it's started to undergo mitosis, but it has not yet implanted into the uterus. It's about 100 to 200 cells, so extremely early in development. There are no tissue layers, so any of those 100 to 200 cells could become any kind of cell. But hematopoietic, hema, is blood. All of these are the blood cells. The next slide has a table. Please do not panic, because I am going to post a table that will tell you exactly which of these cells do you need to know and what do you need to know about it. Essentially, you're going to need to know the cell type and its function. But there are granulocytes because they're grainy. They have cytokines in them that pick up particular kinds of cells, stains. Neutrophils stain with neutral dyes. Eosinophils stain with a red dye called eosin, and basophils stain with a purple dye that is basic or alkaline. They're also distinguished by how many lobes in their nuclei. So this is one nucleus, but it has multiple lobes. The eosinophil has 2 lobes, and the basophil has 1. I am not going to show you slides of these different cells and have you identify them, but that is how they have been identified. Mast cells promote inflammation, and that means they're going to trigger expansion of the blood vessels, and that allows for migration of other cells from the immune system to a site of infection. Natural killer cells might be my favorite name of a cell because their whole point is to go off and kill infected cells. They recognize this MHC, the major histocompatibility complex. It's big, it's complex, and it is compatible with cells. This is what determines your body's sense of self. Which are your cells, which are not your cells. These are the proteins that are used to identify compatible donors for organ transplants. You have to have similar enough major histocompatibility complexes, or the transplanted tissue will be rejected. And it's rejected initially because of natural killer cells. Our natural killer cells have a receptor that is specific to the MHC on our own cells. If it recognizes my natural killer cell recognizes my MHC, it's going to say, oh, well, that's a healthy cell. We're not going to do anything about that. But if I have a transplant and has a different MHC, my natural killer cells will not recognize that MHC as self and would destroy that cell. Likewise, infected cells, they have some sort of activating molecule that binds to a different receptor on the natural killer cell that tells it, pop it, get rid of that infected cell so that that virus can't go off and infect any other cells. So there's our natural killer cell. That lysis happens because of perforin, because to perforate means to punch holes in. So natural killer cells work very similar to the way the complement system has its membrane attack complex, but this is not complement. This is natural killer cells have a receptor to find infected cells. Monocytes, they do not have granules in them, and they become macrophages, which means big eaters. They can completely engulf invading cells or particles. So cells that are damaged and macrophages that have ingested pathogens release cytokines, those immune signals that are pro-inflammatory. They will actually cause inflammation. And inflammation can be healthy if it is at a level enough to recruit the rest of our immune system to help destroy a pathogen. It's not good if it overreacts and over-recruits immune modulators that then damage our own tissues. But this is showing we have damage to the skin, and when that breach happens, we also had an introduction of pathogens, and we will have neutrophils, monocytes get activated. The phagocyte engulfs the pathogen, that releases complement, and that is going to indicate the membrane attack complex should come into play. This slide is showing both a phagocytic cell and things that could trigger reactions in that phagocytic cell. The phagocyte has receptors called pattern recognition receptors, or PRRs, and they recognize things that are associated with pathogens, or PAMPs, pathogen-associated molecular patterns. Those molecular patterns are things that common pathogens have in common, like lipopeptides, short stretches of amino acids with a lipid attached that can be present on either gram-positive or gram-negative bacteria. Flagellin, which is the protein that makes up a flagella, that can be present on gram-positive or gram-negative bacteria. And in this case, peptidoglycan, the cell wall component of both gram-positive and gram-negative bacteria. Any of those molecular components would be bound to a receptor on the phagocyte that would trigger it to bring in the pathogen, fuse it with a lysosome, and destroy it. And here's an image of that. The bacterium gets brought in. It's now encased in membrane called a phagosome. The phagosome fuses with a lysosome that contains enzymes that will lyse the cell. They aren't active until they fuse with the phagosome, and it is now a phagolysosome. That activates the enzymes, they digest the bacteria, any chemicals that are useful to the host cell will be used, and that what cannot be used will be exported by exocytosis from the cell. This poor individual is suffering from leishmaniasis. It is a disease caused by a eukaryotic pathogen, and it is delivered to the host by a bite of a sandfly, and it is evading the tissues in the skin because the bite of the insect broke the skin and introduced the pathogen. That triggers macrophages and inflammation to happen. Mast cells are important in inflammation. We have, again, this break in the skin, pathogens introduced with that, and the mast cells release a chemical called histamine. If you haven't heard of histamine, you've probably heard of antihistamine, like allergy drugs. Again, it's fizzy water, keeping me hydrated. Histamines are a cytokine, a pro-inflammation chemical that cause inflammation. Part of inflammation at the surface is redness, there's heat, but there's also vasodilation. The blood vessels become leaky so that the phagocytes can escape and come out and gobble up the pathogens. A granuloma is a nonspecific innate response where our white blood cells will wall off, form a layer around a pathogen. This happens with many pathogens, but in particular, in this case, it's happening with tuberculosis. The granuloma happens when we have a walled off tissue, all of these little cells surrounding the pathogen. That shows up on a lung x-ray. And the mycobacterium multiply in the macrophages, the very cells that we have that are designed to gobble up and destroy pathogens. The mycobacteria enter, they're in their phagosome, but that does not fuse with a lysosome. And if it's not fused with a lysosome, then it's not recognized by the cell as something to be destroyed, and then it can grow and multiply. This poor individual is suffering from elephantiasis, called that because of the thickening of the lower limbs that is being caused by edema, which is fluid release in response to a worm infection. Again, that is nonspecific. It doesn't know, your immune system doesn't know that it's a filarial worm, but there is an infection, and it is recruiting fluid trying to flush that pathogen out, but because it is large, fluid alone is not going to flush that out. And the worm does have mechanisms that help it to evade detection by the rest of our immune system. So it keeps growing and multiplying and recruiting more irritation. Our hypothalamus regulates our body temperature, but it also can regulate inflammation. So this pathway shown here to get to fever and increase in our body temperature starts with exogenous pyrogen. Exogenous means it's coming from the outside, it's not produced by us. A pyrogen is something that generates fire or heat. So an exogenous pyrogen is something that gets into us and is designed to cause fever, or it will cause fever. In this case, the example is lipopolysaccharide. That's the outer membrane of gram-negatives. There are other exogenous pyrogens, but this is an example. The presence of that lipopolysaccharide or other exogenous pyrogen is a signal to the leukocytes, white blood cells. They, in turn, are triggered to release endogenous pyrogens, chemicals that we produce that will then increase temperature. These abbreviations stand for interleukin-1, interleukin-6, interferon gamma, and tumor necrosis factor. They're all cytokines, chemicals that will trigger immune responses. They all trigger this final protein that tells the hypothalamus, raise body temperature. Raising the body temperature is a nonspecific immune defense. We know that bacterial enzymes have an optimum range. If you go too cool, the enzymes are too stiff to function. You go too hot, you break hydrogen bonds, and the active site falls apart. The same is true for our enzymes. We have a pretty narrow range that they function optimally. So raising our body temperature does risk having our enzymes not function optimally. That's why you feel so crummy when you have a fever. But our bodies do that in the hopes that they can slow down microbial invader metabolism, giving our adaptive immune system a chance to catch up.